So, welcome, good morning. It's 8:30, I know it's early and it's still summer, so I appreciate your being here. But I was looking at the program and it's awesome. I mean, you guys did such a great job of getting sort of the very best speakers, you know, around the world, and a lot of them here in this country, at least according to this program. But I would have loved to participate, you know, to stay for the whole time if I could have. I mean, because it's really outstanding. So you have a broad range of topics that you're going to learn about in this institute. Today I'm going to focus on a narrow one because I think you're going to get a lot of these narrow snippets, and then hopefully you're going to put them all together to form a cohesive overview of what's going on in the intersection of biology and engineering. And in particular, I'm mostly going to talk about cancer nanotechnology today, both in terms of fundamentals on what's important on designing nanoscale probes, as well as when you take it from the design all the way to what it looks like in animal models. So I'm going to focus mostly on what I call these gold nanostars, and I'm going to tell you a little bit about how it can be transported intracellularly, and then finally I'm going to finish with some aspects of our testing in animal models. But I just wanted to express my thanks for this is my research group who's carried out most of the work. Most of the work has been carried out by Duncan, who's hiding behind me, as well as Kayla Culver. Okay, so I just want to get us all on the same length scale, partly because, as we were talking about earlier, there's a range of you guys, well, no physicists, right? And no chemists? Okay, so biologists and engineering, so this is like a perfect slide. Okay, so everything on this left hand of the slide are sort of what you could call self-assembled objects, okay? So you have things as small as DNA to HIV virus particles to red blood cells to human hair to Yao Ming, who's one of the largest self-assembled. Not the largest, but one of the largest. He's very impressive looking if you ever see him in public. Okay, so, but you see the length scales span a great deal. So this is about 2 nanometers in diameter. This is about 100 nanometers. This is about 10 microns, for 5 to 10 microns for a red blood cell, and then you get to human hairs, which are on the order of 60 microns or so. But at the same time, we have the ability to sort of do engineering both at the large scale, which is interesting that we started with a philosophical discussion on transistors, but this is sort of the cutaway of what one looks like, and it's got all these tiny, tiny components in there. And so this takes top-down engineering. And even top-down engineering, you can get three-dimensional structures like this. But if you're trying to make particles or probes that can be used for biological applications, especially in vivo or in animals or in humans, you have to be able to scale them. And so that typically requires us to use some bottom-up techniques. And so some bottom-up techniques can produce things like carbon nanotubes, quantum dots, and gold particles. I show all of these three here because mostly these are the ones that have been pursued in terms of these theranostic or therapeutic and diagnostic applications. So I decided to put part of these nanoparticles on a linear scale. So this is a log scale, and so it's very difficult to appreciate the span over which you need to be able to control things at the nanoscale. So these are structures that are typically used in some cancer nanotechnologies, and so this is a gold nanoshell, and I'll tell you a little bit about its applications in a minute. This is about 100 to 150 nanometers. A carbon nanotube diameter is about 1 nanometer. An average gold colloid is about 13 nanometer, and then a semiconducting nanocrystal, such as cadmium selenide, is on the order of 2 nanometers. And so just as a reminder from what I told you earlier on this slide, right, so DNA is on the order of 2 nanometers, and red blood cells are 5 to 10 nanometers. Other large mammalian cells are on the order of 5 to 10 microns, and other mammalian cells are on the order of 20 microns. Okay, so there's a range of length scales that you might need to span in order to investigate certain biological processes. Okay, so let me just give you some summary of what nanoparticles have been able to do in the context of biomedicine. So there are 3 general applications that most work is focusing on right now. One end is the diagnostics, the ability to detect information both intracellularly and extracellularly. One is the development of delivery agents, and one of them is using these nanoparticles as therapeutic agents. And there are advantages for each of these, and I'll just briefly highlight them. So in terms of, from an engineering perspective, you might care about, if you want to design particles that can fit into these applications, are materials. Materials are very important. And so one of the first questions regarding materials is how biocompatible does the material need to be? It seems to be pretty important, but it might limit the set of materials that you can actually use to design structures. And then secondly, how important is it that it's biodegradable? So should you make all of your particles out of, say, polymers or hydrogels or these types of related organic materials, or is your body going to rebel if you make them out of inorganic materials, such as gold or iron oxide or other types of inorganic materials used today? And the second aspect, besides materials, if you're trying to use these for applications, is where are you going to put what we call the active material? So you need some type of scaffold, or you need some type of carrier, which we call the nanoparticle, but then you have to decide, based on either chemistry or slow release or whatever you want to accomplish, where do you put your agents? And so there are a couple of options. You can put them inside the core, so this might be possible for liposomes or for hydrogel polymer-like particles. Or you might want to attach it to the surface. And of course, you might want to attach it to the surface because you want the particles to get where you want them to go. Okay, so there's all sorts of nice engineering things to consider as we're designing particles for these types of applications. So this is a cartoon that I just highlighted here for Nature Reviews of Cancer. And so their idea is a lot of times people are interested in the ultimate particle, something that can do everything. So for example, this one is very special, right? In cartoon form, it can deliver 2 types of drugs. And it can also give you contrast depending upon where it is located in the tissue. It's got this permeation enhancer so that it can make its way into cells easily. It's got polyethylene glycol on the surface so it avoids unwanted uptake by macrophage. And then it's got these targeting moieties, right? So I show this cartoon because all of these engineering aspects are really, really important, but it's impossible, okay, to put all of them in one structure. And more importantly, if you're using it for a therapeutic, it's impossible for each particle to be identical. So at the end of the day, if you're trying to undergo FDA approval, you have to make everything according to these general manufacturing principles. And if you don't have that, then you're dead. So I'm going to talk about some strategies to simplify the procedure, but also have some potential downstream applications. So let me summarize some aspects of diagnostics. And I'm going to try to highlight some mostly work based on gold particles, but sometimes work based on other material systems as well. Okay, so a colleague of mine, Chad Merkin, had discovered a long time ago, or he had this idea a long time ago, that it'd be really nice to detect specific sequences of DNA. And it would be especially nice if you could just observe it through a color change. So you don't have to have the sophisticated technology of these DNA sequencers, or even the sophistication of base-by-base identification through nanopores. So his idea is, if I have gold colloids and I put a single-stranded DNA, and I put it in a salt solution, or lightly salted solution, they stay isolated. So the color that you have is just red. But then if I design my DNA linker, so half of the strand is complementary to this guy, and half of the strand is complementary to that guy, as soon as they bind, then to form a double-stranded DNA, then the faraway particles are not brought close together. And then that induces a color change, a change in what's called the localized surface plasmon resonance. But more curiously about this type of process, so you could see very specific binding based on color change, is that the melting temperature, or the temperature at which the DNA dehybridizes from a duplex to single strands, is very, very sharp. And so now this is an additional diagnostic that you could be able to use to make sure that you have the specific sequence of interest. So it's a very nice experiment, and this forms the basis for a ton of things that he's doing today, and he has a company based on this. But these are the fundamentals of how it works. He also has this idea of being to detect intracellularly different mRNA sequences. And so he designs this based on a gold particle also, but it's actually quite clever. And so he has a gold particle here. He has an antisense DNA strand, which is longer, and then he has a reporter flare, which is just a fluorescence, which has an overlapping sequence, so it's weakly bound to the particle. But what happens is these particles can be taken up into cells, and then when it finds a target mRNA, so mRNA can be translated into proteins, then this target mRNA has a stronger binding affinity to this antisense strand, and then it releases the fluorophore, or releases what he calls the flare. So if you have the presence of this target mRNA, then you can detect this using fluorescence and flow cytometry, so you know specifically what you have intracellularly. So there's a range of different markers that he has developed for this, and it's being sold right now through Millipore Technologies. So this is sort of another nice example. If you can control the chemistry of the surface, you can actually get out different types of information intracellularly. Okay, so those are two examples of diagnostics, and let me tell you a little bit about, I'll try to combine these a little bit in terms of delivery agents and therapeutic agents. Okay, this is probably one of the most important slides in this entire talk. It's based on this idea of what is necessary to actually get the particle that you designed to go where you want it to go. And it sounds really simple, but it's actually very challenging. And so this guy, Scott McNeil, he's one of the directors of this, what is it called, NCL, National Nanoscale Characterization Laboratory, as part of the National Cancer Institute. And so what they do is they take a lot of these particles that have been developed by individual labs and then run them through the same diagnostic conditions. What are their chemical properties? What are their physical properties? How do they behave in all of these different types of situations? And so what he found is he came up with some set of principles on things that you might need to consider as you're developing particles for both therapeutics and delivery agents. It's not necessarily the same for diagnostics. So the first thing is the zeta potential, or the charge on the particle, is incredibly important. If it's too positively charged, it's going to introduce all sorts of problems into the cellular membrane. If it's too negatively charged, however, then it's going to be cleared out from your system very, very quickly, almost immediately, within less than minutes. And so you want to sort of hit at a zeta potential, or the charge on the particle, close to neutral, or maybe just a little bit. But if it's too negative, it's very bad. If it's too positive, there's cytotoxic effects. Okay, in terms of hydrophobicity, so besides the fact that you might want to put on targeting agents or other things on your surface, if it's too hydrophobic, it's going to be taken up by the macrophage system, and so then it's going to be cleared out in organs like the liver and the spleen, and that's usually not what you're targeting, so that's really bad. And so you mostly want it a little bit hydrophobic, but not too much, otherwise it's going to be eliminated or excreted in organs that you don't want. Okay, so in terms of size, if it's too small, then it's going to be cleared either by the kidneys or the liver. So you want them to be on the order of 30 to 50 nanometers or so. This is rigid. And then if it's too large, it might not make it to within the cells at all. But the good news is, well, not the good news, but the structure of cancer cells or tumors is that they also have a leaky vasculature. So they can often accumulate in tumors if the particles are large by this, what's called EPR effect, this enhanced permeability and retention. So the point of the slide is that you need to consider zeta potential, the charge, you need to consider the size, and you need to consider exactly what's on the surface. And so it's not as simple as the cartoon that I showed you that's saying, let's make this magic particle with all of these different types of properties. Okay, so here's sort of a summary on this. In terms of the diameter, you want it about 50 nanometers or so. If it's too small, it's excreted in the urine. And also, in terms of an engineering or design perspective, if it's on the order of 40 to 50 nanometers, these are about the size of these pits, these clathrin-hooded pits that are important in receptor-mediated endocytosis processes. So this is where this range sort of comes from. You want the surface neutral or slightly negatively charged, and smaller-ish is better, so you have less taken up by macrophages, a process called opsonization. Okay, so here are some carriers for drug release. And there are different types of stimuli to be able to release the drugs. And so there's a beautiful review article in Nature Materials. This is why I put that here. So one type of drug delivery character, and so this is hopefully falling within these design principles that we outlined, are liposomes. And so you can see that they have, it's basically a lipid bilayer that's completely enclosing cargo that you have inside. And so in this case, what they did is they have this amino acid zipper, and they also have doxorubicin inside. Let me go back here. So if you heat this up by temperature, then you're going to force this amino acid to come apart, which then selectively allows the doxorubicin molecules, or these anti-cancer molecules, to be slowly released according to a specific time profile out of the liposome. And so you have to find some way to release it, and this is a very clever way. This is based on temperature. Another example of stimulated drug release using different types of carriers are these porous silica nanoparticles. So these are organic, and porous silica is obviously inorganic. But the idea here is that you can put, you can make these nanoscale pores inside these particles. You can put DNA on the single-stranded DNA on the outside, loaded your drug inside these pores. What you then do is you want to cap these drugs inside the pores until you want to release them, and so you put it with this magnetic nanoparticle that has complementary DNA to what you've put on the edges. So now you can imagine it's all these drug molecules are sort of stuffed in there. And then you use some type of radio frequency, and then you can heat and dehybridize the DNA, and then the drug is released from the center. So this is a very interesting example of, it's actually a little complicated, but it shows you the level of different types of engineering that's necessary to achieve stimulated drug release. Okay, some other examples for drug release. Oh, I think I have. Okay, I guess this is okay. Is again the use of liposomes. So liposomal carriers are the first ones that were FDA approved. Okay, so this whole idea of putting things in terms of small little packages, I think this was about 20 years ago. And so they also decided they put polyethylene glycol on the surface. You have this TAT peptide sequence, and this sequence is very good at penetrating cells. And so they were able to put their drug molecule here, and then as you change the pH, then all of these things can be released. So I show you this example because you can use pH to be able to release drugs from these types of carriers. And then finally, what I'm mostly going to be focusing on for the rest of this talk is the use of gold nanoparticles. And gold nanoparticles are special for a number of different reasons. One of them, they are biocompatible, and their surfaces can be functionalized easily through a gold thiol bond. But also they're responsive to near-infrared light. And so this is the gold nanoshell that I showed you earlier. This is undergoing, I think, it's undergoing clinical trials right now as a photothermal agent, and I'll talk about that in just a minute. But you can use these guys to shine light on them, and then they heat up. The other thing that you can do is you can shine light on these types of particles, and you can heat them, the surface of the particle up enough that you can get dehybridization. So for example, if I have my DNA and I have these other drug molecules around, if I shine light to heat it up, then the DNA dehybridizes at the certain melting temperature, and then I've also achieved drug release. So there's a range of different strategies in order to get these types of things released at the site that you want them. Okay, and then finally is this use as a therapeutic agent. So you might be curious as to why you care about near-infrared wavelengths. And so this is the absorption spectrum of some hemoglobin, oxygenated hemoglobin in water, and you can see it has very high absorption at low wavelengths. So in order for any light-based activation to either heat the particles or to release drugs, you want to be in this near-infrared window. And it typically ranges between about 650 nanometers to about 850 nanometers. So if you can shine light of this wavelength onto your biological system, it can typically penetrate tissue, up to a centimeter or so. So the nice thing about this window also for gold is that gold's optical properties naturally fall into this window. That's another reason why people like gold. And so there's been some seminal work on the use of gold particles as what we call photothermal agents, meaning you shine light on the particles, they heat up, and they destroy the surrounding biological material around it. So one of the first examples was done by Naomi Hallis at Rice University. And so this is the gold nanoshell here. And you can see that it has an extinction spectrum, which is just a combination of absorption and scattering, right in the middle of this, what we call this near-infrared window. So what they did is they took these particles, and they put them on cells, cancer cells, and they shine light on them. And so what you can see is, so this is a stain indicating that the cells are alive. So if you shine the laser only, then all the cells are alive. So the light itself doesn't do any damage or any noticeable damage at the dosages that they use. But then as soon as you put shine light when you had nanoshells on them, then you just killed everything in your laser spot. So this is the first example, which was actually really, really nice. Second example that I want to highlight came out from Sangeeta Bhatia's group at MIT, and this was published here on gold nanorods. But the reason why I like this example, and it goes back to what is important when you're trying to design particles, is what you'll find in the community is everybody finds their favorite particle and they use it to death. Okay? Even if it's not the best particle for their application, they become wedded to it in some strange monogamous way. Okay? But I think it's important as we're trying to figure out what's the most useful approach to designing agents is to be able to do side-by-side comparisons. And so I like this paper because they did a side-by-side comparison. So what they're doing is they're comparing gold nanorods. These are on the order of, say, 10 to 20 nanometers in diameter. They can be 30 to 50 nanometers long. Catherine Murphy here at UIUC is an expert in gold nanorods. And so if you look at the extinction spectrum, you can see that they both overlap at long wavelengths. And for the same gold content, which they determined using inductively coupled plasma mass spectrometry, you can see that the absorption and scattering cross-section of the gold nanorod was slightly larger. But what's interesting is you can look at the temperature profiles. And so if you're shining near infrared light, what's called continuous wave excitation on these solution of the rods and the solution of the nanoshells, that you get a much larger temperature increase for the nanorods compared to the shells. And so when they looked at the photothermal effects in terms of these, putting them onto cancer cells and then shining near infrared light, given that there's a higher temperature response, then you got greater cell death from the nanorods. So it's not to say that nanorods are better than nanoshells in all aspects, but in terms of some of the heating capabilities on a per gold atom level, there seem to be some advantages. Okay, so now what I'd like to do is transition to a little bit of our own work. And I'm going to focus mostly on nanostars, although we work on other types of particles, and I'll try to justify this shape, okay, as we go along. Okay, so we're interested in using these gold nanostars as drug delivery vehicles. So we're not interested in the heating effects. We don't want to shine light on them and have them heat up. What we'd like to do is be able to release a drug from the surface. And so I've told you some of the design criteria for biomedical applications. We want something that's about 50 nanometers, and we also want there to be strong absorption or scattering at near infrared wavelengths. But the problem, if you just want these types of criteria, is that if you have a 50 nanometer sphere, it's going to absorb and scatter at around 520 nanometers in the green wavelengths, which is no good. Okay, if you want the near infrared wavelengths. So all you have to do is to change the shape. So if I go from a sphere to, say, a star, even the rod can do this in some extent, I take away material, then you're going to shift the resonance at 520 to near infrared wavelengths. So this is how we solved this problem. We said, okay, this shape is perfect for getting us into the wavelength range and the size range that we want. Second is to have the surface of these guys be slightly negatively charged. And so our strategy is to use both oligonucleotide ligands, which are negatively charged, but also we need to backfill with polyethylene glycol to mitigate some of that negative charge. And I'll tell you a little anecdote at the end about why we needed to do that. Okay, but in our case, from a research perspective, we're interested in being a little bit greedy. We would say, okay, we don't want just these normal things, but can we get the ultimate in precision targeting? So the first thing we'd like to do is we would like to design cell surface targeting ligands that are also act as anti-cancer agents, meaning we don't want a little Christmas tree particle that has 5 different types of functionalities on the surface, because it's just too hard to get the same number of agents on there, but also the chemistry for attachment and all of these functionalities is different. And so if we can have, say, one molecule that does both, then that's even better. We would like to achieve on-demand release of the drug from the molecules. It takes some time, from the stars. It takes some time for this, what we call a nanoconstruct, to get where you want, both as it circulates through the bloodstream, but also into the cell. So we don't want the drug to come off too early, and we also want it to be able to come off. We don't want it just stuck. And then finally, the ultimate would be able to achieve organelle-specific delivery. And so not just the surface of the cell or the cell membrane, but inside the cell. And so our solution to this is we're using a DNA aptamer. And so aptamers are, you can think of them as chemical antibodies. So they're based on DNA, in this case it's based on DNA bases, but also can fold to bind to a protein in a very specific way with binding constants that are very similar to antibody-antigen binding. These guys can also be loaded on the gold nanostar in really high quantities. We're also using, to release the drug, so we don't want to get heating, or we want to minimize the effects of heating, is we're using these ultra-fast light sources, or femtosecond pulses. So if you have a femtosecond pulse, what's nice is you concentrated all these energy into these bursts, but you can cleave a gold-sulfur bond, for example, and so you get the drug to release where you want. And then finally, in terms of organelle-specific delivery, you can either go after mitochondria or the nucleus, and we just decided to go after the nucleus since it seems to control most of the functionality of the cells. And so at the very fundamental level, my group is interested in what we call nanoparticle-cell interactions. And so this is a cartoon of a cutaway of what a cell looks like. And so we're interested in this type of drug molecule, which is a homodimer, it's a duplex, binding to this specific type of biomarker, or the specific protein. And it turns out this biomarker, which is called nucleolin, and I'll show you the structure. This is the protein data bank structure, but I'll show you a little bit more information in just a minute. It's a surface receptor in cancer cells. It's overexpressed in cancer cells. But the most interesting thing is that it's also a shuttle. So in normal cells, it's mostly in the nucleus, and it's responsible for transport of material into and out of the nucleus. And so in cancer cells, it preserves this functionality, but it's also translocated to the cell membrane. So we might be able to take advantage of all of those characteristics. Okay, this drug that I mentioned is this DNA aptamer. And how it works is that if it binds to this biomarker, then it can start to induce this apoptosis process. So this is BCL2 mRNA is an anti-apoptotic gene, but as soon as you bind the DNA aptamer to this nucleolin, it allows the apoptosis process to occur. So what we'd like to do is take this and surround it with all of these drug agents. Okay, so here's a little bit more detail. The reason I'm sort of pointing this out is because I feel like a lot of times when we're designing constructs, you really don't know the biology that's happening. You just have some idea, oh, I put something on the surface and then I'm going to get it to go where I want. So this is a new marker that has been recently discovered. It's ubiquitous in a range of cancer cells, and I'll show you our own work in that. And it's responsible in ribosome biogenesis and the regulation of cell growth. It also acts as a shuttle between the nucleus and the cytoplasm, and it's not too large in size, and it stabilizes anti-apoptotic BCL2 mRNA. And so this is the drug. You notice that it's a homodimer, and it's not very large. It's about 7 kilodaltons. And it can adopt this three-dimensional conformation where you have these G quadruplexes that can bind to the center of this protein. And the binding constants are pretty good. And so these G quartets, or these G quadruplexes, and they come in sort of this dimerized form in this case, they're very stable. And so unlike normal DNA, which you have it free floating, it'll be degraded by nucleases, these guys are aptamers by themselves are stable. Okay, and so the first studies were done in 2008 using this MCF7 breast cancer cell compared to MCF10A, this normal cell. And you can see that if you treat for up to 96 hours, then nothing happens in the normal cells, but in the cancer cells you get some type of cell death. But you need continuous release or continuous injection over 96 hours, and you need micromolar concentrations. So just to be clear, normally you need about micromolar concentrations for these therapeutic doses of free molecules, chemotherapy agents. Okay, so what are we going to do? What we're going to do is we're going to try to find a biocompatible synthesis to create our drug carriers, because we decided that there's no need to find a very complicated way to make a nanoparticle, and then first of all, it's not biocompatible, and then second of all, it's not scalable. So we used HEAPS, which is this molecule here, and it's actually one of the 12 goods buffers that are used in cell culture. And so these buffers are used in cell culture to maintain pH at a certain carbon dioxide levels to maintain the window. And so there's a range of different goods buffers that you can use, which we have used, but we've just focused on this for now. I won't go through the chemistry on this, but what you can do is you can scale it, and so you can make like 5-liter vats of these guys. This is what a low-resolution transmission electron microscope image looks like, and this is not high resolution, but some zoom-in images of what the stars look like. So they're about 30 to 40 nanometers in size, and they have different branches that come out along the 1-1-1 directions. Okay, and so what we wanted to do is we wanted to test them, their optical properties, and indeed they have absorption and scattering that are on the order of about 800 nanometers. You have to be able to test their size, and so we did this using dynamic light scattering. And I have to admit that, right, these are anisotropic particles. Dynamic light scattering is not going to give you a great size range of these guys, but it's the only way that people are testing sizes en masse. So this is about 25 nanometers, which is what it comes out to be. And then as soon as you add the this DNA aptamer, then it increases by 4 nanometers in radius, and that's what you expect. Okay, so then what we need to do is determine, well, how long does it take, if our hypothesis is that this nucleolin binding to this construct can bring it to the nucleus, how long does it take? And we didn't know. So we did a range of different times of 2.5 hours, 5 hours, 7 hours, and 24 hours. So the blue that you hear is a fluorescent stain. It's a DAPI stained nucleus. And the red here corresponds to a Sci5 label that we put on the DNA aptamer. So we did this so we could sort of try to see where they're situated inside the cells. Okay, so it's obvious by 24 hours there's a ton of particles in the cells, but you don't need to incubate for 24 hours. And so, so we decided, well, this is an intermediate range. These are HeLa cells, and 7 hours looks good enough. And this is the control aptamer. I should mention that I'm not telling you about all of the control experiments that we did, but in biology, there's a lot. Okay, so I'm a physical materials chemist by training, and I do have to say that in order for us to do our biological work, it takes us 3 to 5 times as long, partly because you're actually doing 3 to 5 times as many experiments, although people only really sort of care about what you present. So, but that's underlying all of this stuff. There's a lot of controls involved. Okay, so so one thing that I want to mention here is it looks like if you look at the red signal and the blue signal, there's some overlap. And so you might say, great, it's made its way to the nucleus. But of course, these are confocal microscopy images, and so they don't have the nanoscale resolution that you might need to see whether the particles are actually at the nucleus or not. So what we did is to transmission electron microscopy experiments using these guys. And so in order to do transmission electron microscopy experiments, you take all of these cells and then you pellet them, and then you put them into a resin. And then you need to do slices. They're typically 90 nanometer slices that are involved. And so this is one of these 50 to 90 nanometer slices of HeLa cells. And so I've outlined the nucleus in this red dashed line because otherwise it's impossible to see. But the point here is that anywhere there's these clusters of particles, they cluster into endosomes as they make their way to the nucleus, you see these changes in nuclear phenotype. So you end up seeing these types of invaginations that occur because of the effect of the drug. So what's interesting is that you're using the fact that gold is a very electron dense. So you're using that as a probe, in some sense, an electron probe, electron microscopy probe, but then you're also looking at what's happening on the drug-related surface. So you can visualize these types of interactions. And so if you look at the control experiments, either a scrambled aptamer or different types of normal cells, you see that in the normal cells, the nuclei are sort of these spherical shapes, and it's only, or these egg shapes, and it's only in these cancer cells that you see these invaginations because of the drug. So it's pretty neat. And sort of the first example of what this actually drug does physically. So people have some idea what's happening biochemically, but to visualize this in real time is I think, pretty neat. Okay, so what we then wanted to do is we wanted to use light-triggered release to release these drug molecules from the surface. So we have a femtosecond pulse that comes in, and you release some of the drug. I mentioned some of the drug, which is similarly expressed in this cartoon, because you don't get 100% release of the drug. And the reason for this is the way that the mechanism works. So the light is going to be concentrated in these nanoscale tips. And so it's really only in the region of these nanoscale tips where you're going to get release. And so we did flow cytometry results to show that you indeed get release of the drug inside cells. And then you can do the controls that say the light doesn't affect the cell health. And so there's a couple things that we noticed using this light-triggered release. I guess it's still a little hard to see here. Oh, I didn't change it. Okay, so the first thing is that you still get invaginations if the drug is still on the particle, but if you're able to shine light, and this is what this H-nu stands for, the shape of the nucleus is exacerbated. So the free drug is far more efficacious than the drug on the particle. But you actually needed the drug on the particle to actually get to the nucleus. And so you can see this here. This is one of the first assays that we did. This is a caspase-3-7 activity assay, which is a late-stage apoptosis assay. So how far along is the cell undergoing death? And so you can see if you just have these constructs that there's some amount of apoptosis. But as soon as you shine the light and you release the drug from the surface, then the amount of population of the particles undergoing cell death has increased. What's even more interesting, though, is if you compare the DNA breaks inside the nucleus. And so you really want to do DNA damage. All the DNA is packaged into the nucleus in terms of in a chromatin structure. And so you'd like the free drug to be released and to make its way into the nuclear pore complex and do DNA damage. So if you just have the drug on the particle itself, you notice that, if you're looking at the green, these kind of green lobes, there's no DNA damage inside the nucleus. So you're getting cell death, but you're not actually producing breaks in the DNA chromatin structure. But if you shine the light, you start to see these double-stranded breaks. This is an assay for double-stranded breaks in the chromatin structure. So we have some evidence that as the free drug is being released, it can be shuttled by the nucleolin protein inside the nucleus to do DNA damage, which I think is pretty neat. So basically, you can imagine that the particle is brought close to the nucleus, and then you can release the drug at the doorstep of the nucleus, and then the free drug can go in and do damage. Okay, and so what we did then is we tested the health of the cells. What is the percentage of cell death in these systems? And so we looked at the cell viability. And I tried to reduce the amount of histograms, but the problem is that in biology, there's a ton of histograms. You just can't get away from them. We've tried plotting them in all sorts of different ways, so you can see how we do in terms of your eyes glazing over. But you have histograms, sorry about that. So we compared a bunch of the responses from, we use light to release the drug, the particle itself. We have the controls here, and then we have a bunch of just free drug compared to the normal cells. And so what I only want to focus on here is just what happens to 450 nanomoles of AS1411, which is this aptamer, compared to what happens when it's on the particle. And so if you look at this red hatched versus this purple, you can see that immediately after you've incubated the particles, you have about a 40% cell death. So that's pretty good. If you use this concentration, 450 nanomole, you can see that you only have just a reduced amount of cell death. But what's interesting, if I look over a function of time, so this is one dosage, we're not continuously dosing. After 48 hours, you've reduced the cell death to where about 60% of the cells have died. So you haven't added any more drug. Probably what you're doing is you're doing DNA damage. And so as the cells are reproducing or dividing, the cell doubling time is on the order of 24 hours, then you're just propagating this really bad healthy cells. And so after 48 hours, then the cell death still continues. But the one thing that I want to point out is these are the concentrations of the constructs that we're using, around 0.3 nanomole. And if you can release even just 20% of this near the nucleus, it's over 10 times better in terms of cell death than if it's just freely diffused. So anytime you have drugs that are freely diffused, they can always be degraded by nucleases if they're nucleic acid drugs, like siRNA or single-stranded DNA. Okay, what we were then interested in doing is seeing how ubiquitous this process was. So Nucleolin was recently discovered as a biomarker. And so people only had tested it in 3 or 4 different types of cancer cells. So we decided, okay, let's just test it in a range, in a screen. So we looked at all of these different cancer cell models, and from all of the major cancer subtypes. And you can see that the red overlaps the DAPI-stained nucleus pretty well. And we did all the TEM measurements. But then we wanted to be able to compare the effects of this type of construct in a range. And the other reason to compare in a range of different cell types is that each cell is different. So for any type of tissue-derived drug, you don't have a universal drug. Right? You have something that's specific to pancreatic or specific to breast or specific to prostate. And so what we're actually trying to test here is whether this nucleolin marker, given that it's ubiquitous on a range of different cancer cell types, can it be used as some type of general agent? This is sort of, an academic dream, but it was still interesting to figure this out. So the first thing we did is we looked at downregulation of BCL2 mRNA using real-time PCR. And then, and you can see that if you shine the light, this is the red hatched again, you have a very large reduction for almost all cell types. It tends to be largest in this pancreatic line, as well as some here in this, this ovarian. What we then also did is then we tested the cell viability, so what is the percentage of population cell death or the change in metabolism. And so you can see that for all cases, if you shine light, then these red bars are greatly reduced compared to if you don't shine the light. But the other thing is that if you use high concentrations of the free drug, because we always want to compare concentrations of the drug that's undergoing phase 2. So I forgot to mention that the AS1411 is in phase 2 clinical trials. So we're not inventing drugs. We're trying to repackage and repurpose them. It's far better to put them onto a carrier. And these are all of the normal cell lines that we tested as well. Okay, this is not the last histogram, but one of the last histograms where I think that just sort of summarizes this data. So what I want to reemphasize is that there was a 70% decrease of cell viability by putting the drug on the particle itself. And it's much better than just less than 10% with the free AS1411. And I should mention that this is not the therapeutic dosages. As I mentioned at the very beginning of the talk, the therapeutic dosage on the order of 10 micromolar. So we're not even comparing to this very high amount. We're just using just a little bit. Okay. Am I out of time?
Give it another 5 minutes.
Okay, we did start a little bit, but actually I should probably skip this because we still don't have time for this.
We'd like to have some questions and discussions.
Yeah, okay. So let me just show you if I can find this. Hold on. Maybe I'll just finish up with the in vivo testing. Okay. So as I mentioned earlier, that we're like a materials, chemistry, physics lab. And so there was no way that we were interested in doing animals, except that we had to, right? If you're developing constructs, you have to do animals. So the question is, do we want animals in our lab, or do we want them in a facility? So the good news for us is at Northwestern, there's a facility that houses the animals, and they'll do all the injections and sacrificing and everything else, so we don't have to do it. So I was very grateful for that, because we were so close to having to get these IRB approvals for the lab. And then we had to practice sacrificing the animals and stuff too, so it was good to avoid that. But in any case, it gives you a greater appreciation for some of the studies that you're doing. So we did it in 2 different types of models. One of them is in a nude mice, which has implanted tumors, and one of them is in a rat. And these aren't exactly to scale. So rats are really, has anybody done animal studies here? Okay, so rats are gigantic. They're like this big, right? They're like your little pets that you really care about. And so they're really smart. So if they know that something is going to happen to them, they know it's an abnormal situation, and they bite. And so you have to wear these like steel-lined, or I don't know, these steel-lined mesh gloves to work with them. But the mice, they're super cute. They're like, this big and everything else. And so what I realized as we were trying to do these experiments is we wanted to be good stewards of our models, right? Because not only is it expensive, but it is life, right? What you're testing. So we try to be very careful in this. But let me show you what the results look like. Okay, the first thing we needed to do is we need to be able to scale the drug. Scale the construct. If you can't scale them, it's pointless. And so this is what 5 liters of nanoconstructs look like. And then this is what it looks like when you've concentrated it 100 times. So basically, it's like sludge. There are so many particles. It's very, very thick. But you can still, but the interesting thing about this system is that it doesn't crash. Okay, and so you can still draw it up into a syringe and do tail vein injection. And so what we did is we did a range of different dosages. We haven't reached the maximum dosage yet, but this is a lot of particles. This is 48 milligrams per kilogram of animal mass. And we did this for a range of different, I think 4 or 5 different types. So in terms of the biodistribution, so in the biodistribution, we were looking at there are no tumors in the rats, and so we want to know where are the particles distributed. And so what we did is we did this for 2 days, and then we did this at 14 days. And so what you find out is that, similar to almost all nanoparticles, the liver and spleen like to take up the particles because they're of this specific size. But the thing that's really interesting here, let me see how I displayed it, is that if you look at the accumulation over day 2 and day 14, so day 2 is here, day 14, for the male rats, their response is different from the female rats. And so this is why it's also important to do not just a single sex study anytime you're trying to test an agent. So we had to do both. But in any case, you'll see that the accumulation in the liver and spleen is over 14 days being decreased in the rats. So even though initially there's accumulation there in the male rats, you can end up getting removal from the system. I talked to some friends of mine who were involved in this cytoimmune company, which is based on gold nanoparticles with TNF alpha on there, and they typically clear after 120 days. So maybe you don't want to wait that long, or maybe it doesn't bother you. But in our case, we found that these are rats. The liver is healthy after about 14 days. So the gold particles are taken up in these Kupffer cells, these macrophages. And so sometimes it's good that it's in the macrophages and not in other things, but it's bad in the sense that it's not getting maybe where we want. But this is just the biodistribution with no tumors. And so you can see the stars, what they look like here. We also found that the spleen is healthy after 14 days. So this is the control, and this is at our highest concentration. And so we still see 2 representative cell phenotypes in the spleen. But what's interesting is that the clusters are smaller. So in the liver, they're large. The macrophages just keep taking them up. But here they're much, much smaller. So one of the first studies that we did in these for the biodistribution is the blood work, so the hematology. So we did this for 2 days and 14 days, and I won't go through all of these things in terms of the white blood count and red blood cell count. But the only thing I want to point out is that in this gray area, this shaded area, this is where the range of normal is. So even after 2 days or 14 days of taking up all of these particles, all of these guys are within the acceptable range for both male and female rats. Similarly, we looked at the clinical pathology. So we're looking at all of these amino acid levels. And in particular, these 3 amino acids are associated with liver damage. So even though they might look normal, you want to know, does the chemistry look fine? And in this case, it does. It's all within the acceptable range. We worked with folks at the Developmental Therapeutics Core. This is what I mentioned. They do all of the animal studies. And so if you look at both the liver and spleen, all of these guys look normal according to these stains. And then finally, we're looking at tumor accumulation in mice, bearing both metastatic breast as well as fibrosarcoma tumors. The reason that we chose these models, I just put this here, is both of these, there are no good targeting agents for these types of cancers. This metastatic is also part of what's called the triple negative breast cancer family. And in fibrosarcoma, which is a soft tissue cancer, there are no targeted agents, period. These are rare cancers, so there just has not been a lot of work. So we thought, if Nucleolin is a very good biomarker now, maybe our particles can work there. And so similarly, as you would expect, you get high accumulation in the liver and spleen. Actually, let me talk about this first. But then after 72 hours, you get increase of accumulation in the tumor, which is as good as any nanoparticles that we found in the literature. So nanoparticles, you're always going to lose a little bit to the other organs, but you still get reasonable tumor accumulation. But in terms of fibrosarcoma, you notice that the accumulation is quite low compared to the breast cancer. So this was interesting to be able to test your construct in multiple animal models. But then you also get an increase by putting the polyethylene glycol onto the particles. So this is the lesson that we learned the hard way. So we did all of this engineering to make the particle as good as possible. We loaded like 2.5 times the drug and everything else. But then once we finally did the animal studies, then we realized that just because you have the best drug for in vitro results or cell culture, it's not necessarily the same for in vivo. That was a very painful lesson for everybody involved, but it's important. And now we think about this all the time. Okay, so I'm going to stop there. I was going to tell you some more fundamental work, but I think so we have enough time for questions.
Another 5 minutes or so in this to cover some more fundamentals. In the 5-7 minutes.
Oh, okay. I'll talk really fast. Okay. So as I mentioned, part of my setup was what do we need? What are the design rules that we needed to be able to design either a carrier or a construct to best treat a disease. And the nice thing, since most of you guys are engineers, there's a lot of problems to work on. And so I sort of want to return to that. And one of our hypotheses was that, well, if it's nice that the drug is tightly loaded on the particle, maybe if you increase just by increasing the drug loading, then it can be a more effective agent. So this is what we tried to do. But the other problem that we had found is that, using this process to attach the DNA to the surface takes a really long time. So there was this technique pioneered by Chad Merkin, where you have these gold nanoparticles, and typically they're capped by a citrate layer. This is typically how you make the colloids. And so this whole aging process to load single-stranded DNA takes a long time, but it's necessary because you want to be able to reduce the repulsion between single-stranded DNA, right? Single-stranded DNA is negatively charged, so reduce the negative repulsion between DNA as well as what's on the particle surface is really important. So by adding sodium chloride, this does it. But the problem is that it requires an excess amount of DNA. So 500 to 1,000, up to 5,000 times the nanoparticle concentration. So DNA is inexpensive, but it's not that inexpensive. So you're wasting a lot of DNA. And then finally, it needs 2 days to achieve maximum loading, which is no good. Well, which is fine, but if you're trying to scale it ultimately, then it can be problematic. So there's this paper that came out in Jackson 2012, where they said, okay, instead of this salt aging process, what if we use the citrate buffer at low pH, and then you can reduce both the loading time as well as excess amount of DNA. So we decided to test that with the stars for these two different things. And so what we found out is for the amount of single-stranded DNA, we got the same number of strands, about 180 strands per particle, in 10 minutes versus 2 days. So if you're anybody who's preparing particles, that's amazing. You can run through a lot of different particles instead of waiting 2 days. So what we then decided to do was to apply this strategy to our star, and we found this is the cartoon. So the souped-up stars, you know, they have the asterisks. They have more drug than the less ones. So we again found that the 2-day loading process was reduced to 10 days, which is significant if you're doing experiments. And that the amount of excess DNA, if you're concerned about costs, which we're always sort of thinking about that, was reduced by 1/3. So you save a lot in terms of starting materials. But then here's the thing that was totally surprising, is that the loading was actually increased. So you increase the loading by about 2.5 times. So not only do you reduce the amount of time, these are sort of technological advances, I would say, but then by increasing the loading, it's even more. Okay, so I'll just go through these pictures really quickly. These are confocal images of 2-hour incubations and fibrosarcoma as well as pancreatic cells. And you can see that as you go for 2 hours, as you increase the loading, there's much brighter fluorescence. And similarly for the fibrosarcoma after 7 hours and the pansy cells after 7 hours, it was so bright we almost had to change the gain levels in the microscope. But of course, this information is useful, but it's just fluorescence, right? You need to quantify the number of particles. So if you're ever doing any type of particle analysis, you must do ICP-MS. You have to. Otherwise, you just cannot figure out on a per particle basis how many particles you have per cell. So this is what I want to point out along those lines. So we quantified the number of particles per cell, and it's after 7 hours, and pansy cells is obviously the highest. So the most important thing that we found out is that you got higher efficacy with increased loading. So that was sort of surprising. So what that means is that not only do you have this highly densely loaded drug on the particle, but they're somehow interacting in a multivalent way, which is also a synthetic procedure. It's called multivalent. The multivalent effectors, where you design synthetic ligands where it can bring multiple cell surface receptors together. But in this case, we're exploiting that type of way to get greater efficacy. And so you get superior effects compared to if you have high loading compared to not. And as you might expect, longer incubation times had more cell death. Okay, so I think this is my last. Oh, so let me just go to the summary, because I know we've taken up some time here. So I've tried to give you some overview of what might be involved in designing engineered particles. And so I mostly talked about gold particles. So we focused on our design constraints on optical properties as well as chemical properties. So in optical properties, we want to design things with lots of tips, not only so you can shift the resonance to near infrared wavelengths, but so you can concentrate the fields at the tip for drug release. In terms of chemical properties, we wanted to be able to find a way to do general oligonucleotide loading and also stabilization. If you have free oligonucleotides, they will be degraded by nucleases very rapidly. And then we found that we could also form hodimers of drugs. And so I've tried to introduce this general construct for a wide range of cancer types. And so we're not interested in developing new drugs, but to make the existing ones better and perhaps perform in slightly different ways, which I think I've shown you. And then we're also interested in developing drugs with multiple functions that are still simple enough, not Christmas tree based, that can give you some diagnostic information as well as therapeutics. So thanks for hanging in with me in the first morning session, and I'll be happy to answer questions you might have. Yeah.
Questions. How would you release the drug in vivo?
Yeah, so there are a couple of different ways. So most of the time we're looking to just, so if we're releasing in vivo, that means you have to go after certain classes of tumors that present near the skin. And so, so that's why we focused on the metastatic breast as well as fibrosarcoma, because the mets sort of present near the skin. But also you could imagine using this to clean up tumor margins. And so if you're, in this case, the open areas already are exposed, and you just don't know how far out the cancerous cells are going because you can't see it when you're doing surgery. And so this could provide an additional diagnostic for that.
Great talk, and I really enjoyed that you decided to create a different shape as opposed to spherical or the rod shape.
Mm-hmm.
But I'm just curious, what is the actual percent that remains on the star versus what is released? And if you could change the chemistry to make it more uniform or more of a sphere in the center and make the branches shorter? Or perhaps,
Oh, yeah. Okay.
But can you tune the shape based on the chemistry?
Yes, to some level. So, so the second question, so we can change for sure, you can change like the length of the branches. So for example, this is made through one type of goods buffer, and this one is made through a different types of goods buffer. And so there are 12, I think there are 12 of them. They all work in slightly different ways and they all have different pKa's and things like this. So adjusting how you do the chemistry at the different pH based on the pK of the buffer, that helps. So you can get things that look like this or this. You can also change the relative ratios of the gold starting precursor as well as the buffer. And then we have found that we can tune over the entire visible and near infrared range. So there's ways to do that there. And then in terms of the other thing that you can do, because you notice, I mean, this is sort of full disclosure on all particles, right? There's a distribution in shapes, right? Even within these types of things. So we have been in collaboration with Mark Hersom using this technique called density gradient ultracentrifugation. So what you can do is based on the density, based on the number of branches and things like this, you can separate out in like this large centrifuge tube. And so the different particles will have different bands of color. And so then if you just take that band using a fractionator, then all of those will be very pretty, not monodispersed, but very good in terms of the dispersity. And then your first question was on?
How much is actually released versus what is kept?
Yeah. So we estimate based on flow cytometry results, because we care mostly about what's happening in vitro versus in buffer. But anyway, in cells, it's all about 20% release. So it's not a lot. And so that's based on the fluorescence calibration. But then we could also estimate like how many tips that we have and see whether that corresponds to how many. And we haven't done it. And it's about the same percentage of release even when we have higher loading. So I think what that points to is that the shape matters a lot. You need to get things at the tips. Of course, if we heated the particles up, then you'd get 100% release, but we didn't want to have heating profiles that we couldn't control.
Anybody else?
Are there any specific strategies for using this nanoparticle to deliver drugs into brain? Because brain has the blood-brain barrier. So
yeah, the blood-brain barrier is a curious problem in the sense that, yeah, so there's the barriers, or there's the cells there are different layers, and the separations can be rather tight, meaning even normal molecules, small molecules can't penetrate. So these particles, as well as other types of particles, you do, even just with the normal biodistribution, you do detect gold content using ICP-MS in the organ, in the brain. So somehow it's getting there, and we don't know how. It's not high, but somehow it gets there. A colleague of mine, Chad Mergen, had done this with these spherical particles with siRNA on the surface, and so they were looking at treating glioblastoma. And so, I don't know, they have the same percentage as we do. Like it's this 2%. It's really small that goes across, but somehow it does something. But the mechanism for that is
just curious
unknown.
Whether, they have strategies that could make it more efficient to get into the brain tissue.
But so brain tissue is also very dense. So a lot of times I have friends that work with oncologists at MD Anderson in Houston, and so what they do is they just cut open. Well, I mean, they because it's not too hard to get through the skull, and then you can do all of their stuff that way. It might not be ideal. I mean, they do this in birds and in mice, but I think you can do this in humans too. But I mean, it's just not, it's not ideal. But that's like the most efficient way. It's just like sort of injecting drug. People do this also for testing, although people that really do drug development don't like it, where you just inject into the organ directly. And so, of course, it's going to accumulate there, and it doesn't have to go through the systemic distribution, but I don't know which is. I think people like systemic better.
So basically, I have two questions.
Okay.
So your nanoparticles, you need to do some stability that you will be providing this HEP buffer, right? HEPs buffer.
Heaps is sort of like it's a negatively charged. Yeah. So at the pHs that we're working on, it's slightly negatively charged. Yeah.
So how this molecules bind on the nanoparticle.
Okay. So
do you use any specific chemistry or anything?
we do. So typically, you order DNA. So in the simplest case, you order single-stranded DNA with the disulfide bond, and then you're going to cleave that bond, and then so you're just attaching the sulfur to the gold surface. And so that's typically how we do the chemistry for that. Because the gold-sulfur bond is pretty stable. It's like on the order of, 2.5 to 3 electron volts, so it's not going to come off that easily.
But when you put in the biological system,
What happens? Right.
yes,
Okay.
these bonds will be cleaved, right?
No, the bonds are not cleaved. So typically, what happens? So there are two situations. So when we're doing these studies in vivo in animals, the vehicle is PBS, phosphate buffered saline, and so then you want to know within this buffer, what is the charge on the surface, and it's on the order of -8, -10 millivolts. But then we're doing the cell studies. They're in media, and so in media is complicated because they have all the growth factors for the cells, and it's got salts and all these other proteins. And so there's this thinking right now in the community, which people are trying to get around, is that besides all of this beautiful engineering you've done on this particle, then it's surrounded by something called a protein corona. So all of this non-specific adsorption proteins that bind, and then you've done all this engineering, and then boom, then it doesn't work at all. And so there are some challenges there that people are having in vivo. We haven't really seen a lot of evidence of this protein corona. We've done a lot of different studies, not just in this system, but also in HER2-expressed cancers. And so, but I'm sure it exists. We just don't know whether it's dynamic or it's static. If it's static, it doesn't change, you're done. But I have a feeling it's sort of soft and a little dynamic, which is why you can get targeting at all.
There was a question.
Oh, is this it?
Then we can go back. You want to ask a question?
Yeah, I have a question. I read a paper earlier, I think about 2 years ago, about I'm not sure if it was like a particle like as small as what you guys are working with, but it was something, it was a particle in which they used like magnetic fields to take the particle wherever they wanted to take. And I think they tested it in rats or something. And I was wondering, like, does any of that apply to like nanoparticles at the scale? Like, could you like direct the particle?
Yeah, so you can. So if the particles are too small magnetically, I think, well, it depends on the Curie temperature of the particle also, but they can become super paramagnetic, right? So that means if you apply a magnet, it has a magnetism, and then as soon as you take the magnetic field away, then it all goes random. And so the problem is you can manipulate particles, and we had tried to do this in the beginning by using external magnetic fields to get them sort of to go where you want, but the fields that you need are incredibly high. And so you typically wouldn't want to deal with those types of fields, and they're not as directional as you might like, because you can imagine it's a physical process. So if you have these particles here, you're just jamming them all in one direction. It's not that guided. So I think intellectually, I think the problem is quite interesting, but in terms of practically that's not what people are using magnetic particles for. Usually the ones that are distributed are like iron oxide for like MRI contrast agents. Although my colleague just told me that no more iron oxide-based agents. The FDA just shut down all of the companies. So I was like, oh, okay, that's not good.
Yeah, question in the back. Then we can come back. Yeah.
Okay. I was just kind of curious and a little lost on your in vivo stage experiments, specifically. Yeah. So you said your Aptamer, is that the word?
Mm-hmm. Aptamer, yeah.
Yeah, it was picked up these liver macrophages. What is the consequences of the specificity of where it was picked up, and also why do you think that might be?
Okay, so we first do these. Let me go back. Oh, I can't find it here. Mostly we go. So the first studies we did with rats to look at biodistribution. So what that means is if I do a tail vein injection of these types of constructs, where are they going to go? And so there's no tumor in the rats. So ideally what you would like is if you have a tumor with the animal, then all the particles will go to the tumor and nowhere else. But in the beginning, we just decided to say, okay, we have the stars with aptamers around them. Where are they going to go with no tumor targets? And so at that case, we looked at the distribution in different organs, and in most, and not unexpectedly, they accumulate in liver and spleen. And so, but they're taken up in these Kupffer cells, which are macrophages, but they're not taken up by other liver cells that could induce liver damage. So we tested for liver damage, both looking at TEM, which gives you some general overview of what the accumulation looks like, but then we looked at the blood work, as well as also amino acids, which would be correlated with liver damage, or so they would be elevated, for example, and we didn't see any of that. But then we also looked at slices of the organs in these what's called H&E stains, and that all looked normal, like there were no abnormal modules. So my conclusion from this is, okay, yes, you're going to get uptake in these types of organs, and then the challenge is how much can you reduce the uptake in those guys versus accumulation once you have a tumor implanted. It's not an easy type problem. It's a universal problem that everybody sort of is trying to work through right now.
There's a question from there, and then go ahead.
What about the optimal state of potential for nanoparticles to cross the cell membrane?
Yeah, so in the beginning, everybody thought you needed like positively charged particles, cationic particles. And then there was a whole spate of work that said, oh, for whatever reason, these highly negatively charged particles can cross. But some of the hypothesis of that was that you're binding multiple sites on the cell membrane, and so that's helping the uptake process. Whether it's endocytosis based, I'm not sure, but it's helping the uptake process. But then what people found in terms of in vivo work, so not for cell work, but for in vivo work, you wanted them close to neutral, because if they were too negatively charged, they would be just taken up by macrophages. This is what we found too, is bad. But if it's too positively charged, then you introduce all of these cytotoxic effects that were sort of unknown. And so then I think empirically people, combining all of this information together, they said slightly negative to neutral is best. This is why they add polyethylene glycol, to sort of make things a little bit more positively charged. But they can still work. It's just a matter of, are you going to lose things to different organs unnecessarily or not?
Okay, last question.
So you mentioned some histology work. Just wondering if you look at histology of the tumor cells, that it was millimeter thick, and you have the free drug versus the drug nanoparticles. I have a feeling that it's going to diffuse much slower than the drug itself. So if you look at histology, does the drug distribute uniformly across the tumors, or is it just like the surface that next to the blood vessel?
Oh, that's a good question. So it's very difficult for us to look at it's difficult for anybody to look at free drug diffusion in tumors. And so typically the models for that is that free drug is going to have to be next to a marker, like a fluorescent marker, like luciferase or something like this. And then you can see maybe what the diffusion profiles look like. In our case, we don't know whether you have the particles that are just close to the blood vessel on the tumor, or whether they're distributing throughout. So that's sort of the next thing that we need to do, because maybe you're referring to this. There's this recent paper by Yunan Shah at Georgia Tech, and he found out that he looked at the comparison of particles. They were untargeted particles, but particles of different shapes. And so he found out that there were different, and he'd use radio labels to look at, not ICP-MS, but radio labels to look at where things were. And he found out that different shapes could have different penetrations compared to ones that were, say, close to the blood vessel. So we don't know that, but my suspicion is that we can actually penetrate pretty far based on some other results that we've done in sort of three-dimensional cells, these human tumor neurospheres. The star shape, or I don't know what's going on though, but it can penetrate to the center while the spheres sort of stay on the outside. But that's the best evidence that we have for that.
Okay, let's thank Professor Teri Odem, and as a symbol of our appreciation, there's a little gift for you.
Oh, great. Thank you.